Welcome to the deep dive. We're tackling a fascinatingly complex parasite today, Toxoplasma gondii. You all wanted us to really get down to the nitty-gritty with this one, huh?
Oh, absolutely. We've got some seriously in-depth research articles here diving deep into Toxoplasma gondii in dogs, PhD level stuff.
Yeah, we're talking cellular and molecular mechanisms, definitely a deep dive. So let's do a quick overview for any veterinarians out there who aren't living and breathing toxoplasma research every day. I'm looking at this study, identification of immunogenic proteins within the dense granule proteins. It seems like we're really getting into the weeds of immune responses here.
We are. This research is all about the tachyzoite stage of Toxoplasma gondii. So in the acute phase of infection, these tachyzoites, they're multiplying like crazy. And what they do is release proteins, dense granule proteins, or GRAs. And these GRAs, they're essential for how the parasite interacts with the host's immune system.
GRAs, okay, so like the parasite's secret weapon to avoid being destroyed by the immune system.
Exactly. Some GRAs can actually hijack the host cell's own machinery. It's how the parasite hides, sets up shop and creates a long-term infection.
Sneaky.
This particular study, they used this really cool technique called immunoproteomics. And basically, they wanted to figure out exactly which GRAs the host's immune system recognizes.
Immunoproteomics. That sounds intense. Break it down for me.
All right, so imagine this. They take blood serum from dogs that were experimentally infected with Toxoplasma gondii. Right?
Yeah.
This serum, it's got antibodies, which are like the immune system's memory bank of past infections. And then they expose the serum to a bunch of toxoplasma proteins, including those GRAs.
Right.
And the antibodies in the serum, they'll bind to any proteins they recognize from the earlier infection.
So it's like a most wanted poster for the dog's immune system.
Exactly. By seeing which proteins bind the strongest, they pinpoint the most immunogenic GRAs, the ones that really set off the immune system's alarm bells.
So kind of like finding the parasite's weak spot, the Achilles heel.
That's it. And get this. Figure 1 in the study, they've actually created a visual of these immunogenic GRAs. Each bar on the chart, that's a different GRA protein, and the height shows you how immunogenic it is. Taller the bar, the stronger the immune response.
Oh, wow. Yeah, some of those bars are way up there.
Right. And what's really interesting is that some of these highly immunogenic GRAs, they were unknown before. We're still learning so much about this parasite.
It really drives home how much we still don't know. Now, the study keeps mentioning this humoral immune response. Can you give us a quick refresher on that?
Of course. The humoral immune response is all about antibodies, those Y-shaped proteins we were talking about. They're made by special white blood cells, B cells. And when an antibody latches onto an invader, like our friend the parasite, it's basically waving a flag, signaling other immune cells to come destroy it.
So it's like sending out a distress signal.
Exactly. Now, this study focused on the humoral immune response to Toxoplasma infection in dogs, but there's another important part of the immune system we can't forget about, the cell-mediated immune response.
Yes, the T cells, right? Like the frontline soldiers of the immune system.
That's them. T cells can straight up kill infected cells, which stops the parasite from multiplying and spreading. And they also kind of direct the overall immune response.
So you've got the humoral response with the antibody flags and the cell-mediated response with the T cell soldiers all working together to keep Toxoplasma in check.
Exactly. This study mainly looked at the humoral response, but understanding both parts of the immune response is super important for creating effective vaccines and treatments.
Absolutely. Another thing they used in this study was Western blotting. Now, I vaguely remember that from my days back in the lab, but could you remind us what that is again?
Sure. Western blotting is how we find specific proteins within a mix of tons of other proteins. Think of it like a bowl of soup. You've got all these ingredients, but you only want the carrots. Western blotting is like picking out all the carrots and ignoring the rest.
Oh, okay, I get it now. So they use that to confirm those highly immunogenic GRAs they identified earlier.
Exactly. They separated the toxoplasma proteins by size, put them on a membrane, and then used antibodies specifically made for each GRA to see if they were there. If you look at figure 2, you'll see these bands. That's confirmation those key GRAs are present.
It's amazing how much detail they get into.
I know, right? And this detailed look at how the parasite messes with the host's immune system, that's what's going to lead to big breakthroughs in how we prevent, diagnose, and treat this.
Totally. This is already fascinating, and I have a feeling we're just scratching the surface here. Right?
Oh, absolutely. This study was just one piece of the Toxoplasma puzzle. There's so much more to explore.
So before the break, we were getting into those sneaky GRAs on the surface of the parasite. But this next study, the role of microRNAs in Toxoplasma gondii infection, takes us down a totally different path. We're talking next level stuff here.
It really is. We're moving past proteins, going even smaller to microRNAs.
Okay.
So these microRNAs, they're like tiny molecules acting as genetic dimmer switches. They fine-tune how active our genes are. And it turns out Toxoplasma can mess with these microRNAs to help itself out.
Okay, hold on. I'm getting flashbacks to genetics class, and I'm not sure that's a good thing. Can we rewind a bit? What exactly are these microRNAs, and why should we care about them when it comes to Toxoplasma?
All right, so imagine our DNA as this massive library, right? And it's full of blueprints for building proteins, which do all the work in our cells.
Okay, I'm with you.
But we don't need all those blueprints active at the same time. That's where microRNAs come in. They're like the librarians, deciding which blueprints get used and which ones get put back on the shelf.
So they're controlling which genes are turned on or off, or like how much they're being used.
Exactly. Gene expression. And the really wild part is that Toxoplasma has figured out how to hijack this system. It can release its own microRNAs into the host cell, or even mess with how the host makes its own microRNAs.
So Toxoplasma walks in and starts telling the librarians what to do, causing chaos in the library, messing up which proteins get made.
Perfect analogy. And when normal gene expression gets messed up like that, it can really impact the host cell, making it a much better place for the parasite to hang out.
And this study specifically looked at microRNAs in the brain during Toxoplasma infection, right? Something about astrocytes.
Yeah, astrocytes. Think of them as the support staff for the brain. They bring nutrients to the neurons, clean up the waste, even help regulate the immune response there.
Right. Important cells.
And this study showed that Toxoplasma infection can actually change the expression of microRNAs in those astrocytes. Disrupts their normal work, you see.
So the parasites messing with the brain support staff, that can't be good. What kind of effects did they find?
They saw some interesting changes, like Toxoplasma infection caused a drop in a specific microRNA called miR-146A in the astrocytes. And we know this miR-146A, it helps control inflammation in the brain.
Hold on. So by lowering this miR-146A, Toxoplasma is essentially saying, more inflammation, please.
Pretty much. And more inflammation in the brain, that's not good news. It can damage neurons, mess with normal brain function. Could be a reason why we see those neurological symptoms with toxoplasmosis.
It all makes sense, but it's kind of scary how this parasite operates. I'm looking at figure 3 in this study, and they use something called quantitative PCR. Could you remind us what that is again?
Of course. Quantitative PCR, or qPCR, it's like a molecular copy machine. We can measure how much of a specific RNA molecule we have in a sample.
Okay.
So in this case, they used qPCR to see the levels of different microRNAs in those astrocytes.
So like counting how many of each microRNA librarian they had in those astrocytes.
You got it. And the results were pretty striking. They found Toxoplasma infection really changed the expression of a lot of different microRNAs in those astrocytes, not just miR-146A.
So it's like a multi-pronged attack on the brain using those microRNAs to subtly but effectively manipulate gene expression in those astrocytes.
That's a great way to put it. And when you disrupt normal astrocyte function like that, it can have pretty big implications for brain health and behavior during Toxoplasma infection.
This is mind-blowing stuff. We've gone from GRAs on the surface of the parasite to these tiny but powerful microRNAs messing with gene expression in the brain. Really shows you how complex this parasite is and all its tricks.
It does, doesn't it? And it makes you realize how important it is to understand all these molecular mechanisms if we're ever going to stop Toxoplasma infection or treat it effectively.
For sure. You know, we've talked about GRAs, we've talked about microRNAs, and just when I thought we covered all the bases, this last study throws us a real curveball. It's called Modulation of Gut Microbiota by Toxoplasma gondii in mice. And it's a whole new ball game.
Right. We're going beyond the parasite itself here. We're talking about the gut microbiome, that whole universe of bacteria, fungi, viruses, all living it up in our intestines.
I'll be honest, when I think Toxoplasma, I think brain, maybe muscles, gut microbiome. Not exactly the first thing that comes to mind. So why are we even talking about this?
Well, it turns out that gut microbiome isn't just about digestion. It's way more than that. It plays a huge role in our immune system, our metabolism, even our behavior. And we're starting to realize that a lot of parasites, Toxoplasma included, can actually change the makeup and the function of the gut microbiome.
So Toxoplasma is like that house guest who not only overstays their welcome, but also rearranges your furniture.
That's a great way to put it. This study, they looked specifically at how Toxoplasma infection affected the gut microbiome in mice, and they used a technique called 16S RNA gene sequencing.
Okay, hit me with the genetics knowledge. What is that exactly?
Basically, it's like taking a head count of all the different bacteria in a sample. Like, imagine you walk into a crowded room, 16S RNA sequencing tells you who's there.
Okay, so they're figuring out who's who in the gut bacteria world.
Exactly. And by comparing the guest list, so to speak, between infected and uninfected mice, they could see what Toxoplasma was doing to the whole community.
And what did they find? Was Toxoplasma playing favorites, inviting some bacteria in and kicking others out?
Exactly that. They found that Toxoplasma infection actually changed how many of certain bacteria were in the gut. Like they saw more bacteria from this group called firmicutes and fewer from this other group, Bacteroidetes.
Those names sound a bit like something out of Star Trek. And
Yeah.
seriously, are these changes good, bad, just weird? What does it mean?
Well, the ratio of firmicutes to Bacteroidetes, that's actually really important for gut health. When that ratio is off, it can lead to a whole host of problems: obesity, inflammatory bowel disease, even neurological disorders, you name it.
So by messing with this delicate balance of bacteria, Toxoplasma could be affecting the host's health in ways we never even considered before,
Yeah.
beyond just, the parasite itself.
Exactly. And it's not just about who's there, but what they're doing. The gut microbiome produces all these metabolites. Think of them like chemical messengers that affect different bodily functions.
Okay.
And Toxoplasma infection can change the production of these metabolites too.
So not only is Toxoplasma inviting the wrong crowd, but it's also messing with the drinks.
Another great analogy. This study found that Toxoplasma infection actually changed the levels of these things called short-chain fatty acids.
Okay.
And these fatty acids, they're important metabolites produced by the good gut bacteria. And they're involved in controlling inflammation, immune function, even brain health, believe it or not.
Wow. Okay, so this is kind of alarming, right? We've got Toxoplasma affecting the brain, manipulating the immune system, and now it's like it threw a wrench into the gut microbiome too. Does this parasite impact the entire body?
You're not wrong to be a little freaked out. Toxoplasma gondii, it's incredibly adaptable, and it can influence its host in some really subtle, complex ways.
So where does that leave us? We've uncovered all these complicated mechanisms, but how do we use this knowledge to actually protect animals and people from Toxoplasma?
That's the million-dollar question, isn't it? But these discoveries, they open up all these new doors for research. Can we find ways to manipulate the gut microbiome so it's not so easy for Toxoplasma to take hold? What about targeting those specific microRNAs to stop the parasite from setting up shop in the first place, or even changing host behavior? It's all on the table now.
It's crazy to think about,
Yeah.
these tiny organisms, parasites, bacteria, even viruses. They can have such a huge impact on our health, even our behavior. Makes you realize how interconnected everything is.
Absolutely. And it just goes to show how important it is to keep digging deeper, keep asking questions, and keep trying to understand this intricate dance between hosts and the little worlds living inside them.
Well, listeners, that wraps up our deep dive into the world of Toxoplasma gondii. It's a fascinating and sometimes unsettling world, but hopefully you've learned a thing or two today. And hey, who knows what we'll uncover next?
Yeah.
From Vet Neurojar. Keep those minds inspired, hearts light, and tails wagging.